Do you believe that evolution is fair or unbiased? Scientific studies show that if you have adversity in your early life, your brain development may hamper. But the problem is it does not affect male and female similarly. So today we'll discuss an article that explored these sexual differences during brain development and the reason behind it. During our conversation, we'll talk about this article, whose title is Early Adversity Causes Sex Specific Deficits in Perforant Pathway Connectivity and Contextual Memory in Adolescent Mice. So, to discuss this article, I have Rafiad Islam, the first author. Rafiad Bhai, welcome to this Research Digest podcast.
Thank you so much for having me.
So Rafiad Islam is currently working as a associate research scientist at Yale University. So before going into your particular research article, please share your career journey and academic journey with us a little bit.
Thank you so much. I did my undergrad and master's degree from biochemistry and molecular biology in University of Dhaka. After my master's, I worked around 1 year in ICDDRB. After my research career in ICDRB, I joined as a lecturer in Maulana Bhashani Science and Technology University in Tangail in Department of Biotechnology. I worked there for around 5.5 years, and I promoted to assistant professor over there. And then I went for my PhD journey to Japan as a Monoboshu scholar. I did my PhD from Shimane University, Japan, which is a small university near the Hiroshima. So in Shimane University, I did my research on theobromine improves working memory by activating a pathway, which is CAM kinase 2, CREB, and BDNF. And this is on rat as a model system. So that was my PhD journey. After my PhD, I came to US as a postdoctoral associate in Yale University. And I was working in a Department of Psychiatry and as a postdoctoral associate. And I spent 4 years as a postdoc in that department. During this time, actually, I was working on a few different projects. And one of the project is like the project what we are going to talk today. And that is like how early life adversity affect the brain development. And
wonderful. Thanks for sharing with us. So you had a long journey even before joining this current position. You worked in a non-profit research organization, which is ICDDRB, which is kind of the one of the most prominent research organization in South Asia. And then you worked in an academic settings as a assistant professor, and also then you did your PhD. So that's a long and wonderful journey. So the pathway you worked on, are those associated with neurological diseases, and what kind of diseases?
Yeah, this is a very interesting story of my career. So actually, I started with microbiology. So then when I was teaching in the university, so neuroscience is one of the fascinating part of the teaching for me. And after that, I found that one of the university that I applied for my PhD in Japan. So that lab actually is not a neuroscience lab.
Okay.
That is mostly a physiology department,
Okay.
but they started working on neurology. So they work on Alzheimer disease and different kinds of neurodegenerative disease. Yeah,
Okay. Okay. Wonderful. That's how you got into this current
yeah,
lab and these research, interesting research projects. Okay,
yeah.
that's
Also,
wonderful.
also just to add,
Sure.
after my postdoc, actually I moved a little bit more. So now I moved to a different department, Department of Neurology,
Okay.
where my research focus is little bit changed from my previous one. So now I'm working on Parkinson's disease.
Okay.
So it's a lot of changes in the career. Yeah.
Yeah. Okay, that's wonderful. So let's go into your particular research article,
Hmm.
which was published in Biology of Sex Differences Journal by BMC.
Hmm.
So to understand your paper, we need to understand some of the backgrounds.
Yep.
So give us some idea about previous studies that actually linked human brain development with childhood adversity.
Okay. So yeah. First, actually, I want to explain a little bit about what is child adversity of early life or early life adversity.
Sure.
So yeah, so there is a big study is from CDC and Kaiser. So Kaiser is a, like a non-profit organization who do some kind of methodological studies. So they made a big studies. It called ACE studies, like adverse child adversity. So on that study, so they found that 1 out of 6 adults experience like 4 or more different types of adversities in their life, and at least 5 out of 10 that cause the death and associated with different types of diseases. So, and the adversities may be like neglect in the early childhood, maybe kind of bullying, maybe sexual abuse or emotional neglect or extreme poverty. But like in all this problem leads to develop different kinds of psychiatric disorders like depression, anxiety, like substance abuse,
Mhm.
PTSD or psychosis, different types of neurological disorders.
Okay.
So, a few of the labs, actually, very few labs working on this early life adversity. One of the prominent lab, those who are working, is from Harvard Department of Psychiatry, like Tiger and Samsung. So they are mostly working on this early life adversity for last 20 years. So and like the Taser, he is like a PhD from Yale and then moved to Harvard for his career.
Okay.
And so he developed different kinds of adversity models and used as a replica for rodent model as human.
Okay.
So in our background of the study, like we in my previous lab, we used to work on different kinds of adversity models. So the model we are working here is kind of written, said as LB. It's kind of in the abbreviation is limited bedding. So what happened, like in the mice, like usually you have to give a standard cage, like a 500 cc of corn cob bedding and with a nestlet, and then put the mice. But in case of LB, we used to put like 125 cc of bedding, and there is no nestlet. So the paradigm start in the early time point, like when the mice is pregnant. So we move the mice into the maternity cage. So
Mhm.
we set up that maternity cage like in control and the LB condition. And on that condition, the mice give birth.
Okay, okay. So this is about limited bedding, that the method you used
Yeah, yeah,
to
yeah.
study this one.
Hmm.
Okay.
So for the study, you are asking like what kind of study before people do about
Yeah,
this.
yeah. And also I'll just tag a little bit.
Yeah, yeah,
Another question. What was found before you started this
yeah,
study? So, what was found about the differences between male and female in terms of their early life
yeah,
brain development?
yeah. So actually, this is the first study that actually take account male and female in case of this adversity in the adolescent time point.
Okay.
There are few studies before that, like around early stage of the life, like P2 to P9. So that time point, some researcher from that Harvard and some researcher from Stanford. So they work for this kind of studies, but not too many studies that are focused on the sex differences.
Okay.
Mostly, all the studies are mostly male prominent. So the reason behind this, like in our few studies, we are seeing that in the behavior study, we see a big difference in male and female. So that actually intriguing us to look into it, like why there is a difference in the behavior in male and female, and is this happening from the very early
Mm.
stage of the life or like in later time point it's happening? So to
Okay.
find
Okay.
out that, we started these studies from the like early time point.
Okay. Got it. That's interesting. So first you observed the differences in behavior,
Mm-hmm.
then you decided to go into
It's,
mechanisms of
it's a
why
kind of
what's happening.
backward study actually. Yeah.
Okay. Okay.
Usually in neuroscience field, we just first want to see the phenotype in the mice. If we find something
Mhm.
interesting there, then we go back in the molecular mechanism.
Okay. Okay. Got it. So let's go into your major findings. So
Uh-huh.
you already mentioned about limited bedding, and
Mhm.
this is a particular model of mouse or rodents. So
Mhm.
tell us a little bit more about that,
Yeah.
and also why did you choose this method as the early life uh, early life adversary model?
Okay.
So, is there any other method available as an alternative to this thing?
Yeah. So this limited bedding or LB, or another name is there, limited bedding and nesting, LBN. This is a well-established paradigm in rodent model for
Mhm.
early life adversity because it mimic a environmental deprivation by reducing the nesting material and bedding.
Mhm.
This is kind of, you can like say in human, it's kind of a poverty.
Poverty.
Yeah. So, and leading to a fragmented or unpredicted maternal care. This is kind of, you can say, neglect in child raising. Like what happened in limited breeding maternal care. So they have a very fragmented maternal care because, like, every time the mother is very anxious about their kids, about their pups, that they are not getting enough care. Because like the if you see in the like the cage, there is very small amount of bedding, and they
Hmm.
made a nest very at the corner, and the pups like mothers are like taking the pups every time, like in the side here and there. So it is a not a consistent care of the mother. So we say that as a fragmented or unpredicted maternal care,
Okay, okay.
which is
Right.
very critical in development uh, window, like around, if this is happening in early stage of the life, like P2 to P9. So this time point is very crucial for the brain development. So
Right.
in this limited bedding model, so it is happening. There are a few others early
Okay.
life adversity model, like maternal separation, like
Hmm.
nested care. So in maternal separation, pups are separated from the moms for like 1 hour, 2 hours, and we checked that model. So it is very like robust model. Like child, like the pups are crying. So
Wow.
you can actually measure the intensity of the pup crying. So we have that system in our previous lab.
Hmm.
We can record that, and when there is a baseline, like when they're with the mother, so there was no crying.
Okay.
And in the time, like when they are crying, their corticosterone, that is the stress hormone, is very
Hmm.
high.
Okay. That's interesting. So, this limited betting is kind of an indicator of early life adversity. We can say it is indicator of human
Yeah,
adversity as well. So,
yeah.
if we compare this to lack of food, lack of
Mhm, mhm.
proper
Yeah,
household,
yeah.
yeah, maternal care,
Maternal, yeah,
maternal care, those things.
yeah,
So, yeah, okay, it is well established. Okay,
yeah, yeah.
that's wonderful.
Yeah. So, and the time point we are using for this study is another crucial thing,
Okay.
because most of the model they work on like P2 to P9, this time point as
Hmm.
a limited bedding, but we extend this time point until P25,
Okay.
which is like if I want to give you an idea with the human development with the mice development,
Hmm.
like 2 to P9, this time point we can say like gestational last trimester to 1 to 3 years of age.
Okay.
And if I extend it to P25, that can be extend in human like adolescent time point.
Okay. Okay.
So we extend this time period for a longer time point just to see what is happening in the adolescent time point.
Kind of to see the long term effect.
Long, little bit long term, like
Hmm.
you can say like intermediate time point.
Okay.
Yeah.
Okay. So that's interesting. So there are several very crucial findings in your study.
Mhm.
Uh, so first, tell us about your findings on myelination in the hippocampus and
Okay.
kind of why is it prominent in male mice?
Okay. So before going to that, I will give you some brief idea about how we start the experiment actually.
Yeah, sure, sure.
Yeah. So uh, to get an unbiased approach, so we just started with RNA-seq
Hmm.
from the hippocampus of the P17 pups.
Okay.
This is the in between P25 to P0, P17. So the reason for choosing this specific time point is like in this time point in the development of mice, there are a lot of things happening in this time point.
Which day of their life period
Okay,
is
so it is the
P17?
17th day after birth.
Okay, that's what you mean, P17.
Yeah,
Okay.
P17. P1, P0. P0 is like the day it born, and
Okay.
P7 is the 17th day of the
So the
life.
mice. Yeah, okay,
Yeah.
like sure. Okay.
So on that P17 days, actually in the developmental process, many things happening like in this time point, like the dentate gyrus of the hippocampus, it is formed in this time point, and
Hmm.
connectivity of the entorhinal cortex. I will explain what is entorhinal cortex a little bit later. So like just to
Okay.
give you an idea what are the different specific things is happening on that time point.
Hmm.
This is this by this time point, P17. This like the connectivity and the dentate gyrus formation is almost done. So
Okay.
after that, if you want to do something, it's like not plastic. So
Okay.
it is like done. You cannot do anything.
Okay. So that's why you chose that time point
Yeah.
for RNA
There are
sequencing.
few other things that just
Mhm.
started, like synaptic pruning, that are in the peak. Synaptic pruning is a very important process in brain development, because like when we like born, in our brain, lot of neurons are
Hmm.
there. Those are not all needed for our purpose or our existence. So the
Okay.
microglia eat those extra neurons. That is called a synaptic pruning. So they clean up the extra things. So this is the time point where the synaptic pruning is in the peak point. And few other just started, like the myelination of the brain,
Hmm.
different part of the areas of the brain start myelination. So that's why we choose this time point for the RNA-seq analysis, so that we can get an idea what is happening in the RNA level. And interestingly, in the RNA-seq analysis, when we did a pathway analysis of the RNA, and we found that the myelination related pathway is the highest downregulated pathway in LB
Okay.
compared to control.
Hmm.
And when we
That's your
look
observation from RNA-seq
for
analysis.
RNA-seq, and when you look into the pathway analysis, I mean transcription factor analysis,
Hmm.
we found that the transcription factor like the MIRF, the OLIC2, the SOX10, these are the transcription factor related with myelination.
Okay.
These are highly downregulated in LB.
Hmm.
And when we check for the different kinds of gene, and we found that the mature oligodendrocyte related gene like the MBP, then PDGRF1, the MOG, MAG. These are the like mature oligodendrocytes. So give so I'll give you a little bit more idea about the neurogenesis. So from the neuronal stem cell, we will have a intermediate situation of the neuron that is called OPC,
Hmm.
oligodendrocyte precursor cells or progenitor cells, and oligodendrocyte progenitor cells turn into mature oligodendrocyte. These are the main three part: neuronal stem cell, oligodendrocyte progenitor cells, and the oligodendrocytes.
All right.
So what
Okay.
we find, there was not that many change in the mature oligodendrocyte related gene, but there is a big change in the oligodendrocyte precursor cells, and that is a very like highly. I mean, there is very low amount of oligodendrocyte precursor cell in the LB mice. So getting that, we want to see like what is happening there. Then first, what we did,
So at this point,
yeah,
I should say,
yeah.
at this point, by LB mice, do you mean both male and female or just male?
So actually, in this time point, you cannot distinguish male female that clearly.
Okay, okay.
So what we take, like we took like all the male female. I mean all the leader mates.
Mm-hmm.
So there is no distinction in this time point. But like little bit later, when we like after P17, around P17, P18, you can see actually little bit
Mm-hmm.
change in the genital and also in the female mice. So you can distinguish after around P17, P18.
Okay,
So what we did, we collect some of the brains in P17. We did the RNA-seq and few brains we stain that with MBP.
okay.
MBP is a marker for the myelination.
Mm-hmm.
And what we see, like around the hippocampus, there is a area in between CA1 and CA3. CA1 and CA3 is like two different areas of the hippocampus. So
Okay.
hippocampus is like a kind of a banana shaped thing.
Mm-hmm.
So CA1 and CA3 is like on top it is CA1 and on down it is CA3. So in between that area, we found a specific area that is highly myelinated, and
Mm-hmm.
that area is called striatum lacunosum molecularis.
Okay.
So in short form, it is SLM. And our
Mm-hmm.
striking findings, like these specific area in LB and the compared to control, there is a huge difference.
Okay.
Here in LB, there is a very low amount of myelination, and in control, there's a like regular myelination. So that
Mm-hmm.
means like it support our RNA-seq data that there is a difference in myelination in the hippocampus.
Okay. Okay. So, yeah, myelination is different uh, with the treated LB treated mice and
Mhm.
compared to the control group. But
Mhm.
tell us a little bit about the importance of myelination. Why myelination is
Okay.
important?
So myelination is called as a wrapping of the neuron.
Hmm.
So in the age time point, like in the developmental time point, so the myelin precursor cells, like the OPCs, like the a, oligodendrocytes, it comes closer to the neurons and it wrap up the neurons. So the reason for myelination, I mean, what is the purpose of myelination? So myelination increase the conductivity of the neuron, like the signal that pass from one area to another area or any part of the body, it need to be very fast. So myelin makes this process faster by a solitary movement. You can say like there is a node of Ranvier in the neuron. So node of Ranvier means like there is a small opening of the neuron,
Hmm.
and there is a kind of, you can say, is a rubber wrapping on the neuron, and there is a small gap. So the
Right.
signal actually travel through solitary movement, like from one gap to another gap, one gap to another gap, one gap to another gap. So myelination actually helps this process to make it faster. If there is no myelination, so the signal have to
Okay.
reach from A to B like 10 milliseconds. But when there is myelination in this like 1 meter length, so it will go in 1 millisecond.
Okay, okay. So it kind of makes our neuron more functional and active
Much
and
more
efficient.
active. Yeah.
Mhm.
Yep.
Okay, thank you. Yeah, so and in your imaging experiment, one observation was that this myelination is different in the hippocampus region
Yep.
in male mice compared to female mice, right?
Female mice. Yep.
Tell us a little bit about that.
Okay. So as we see, when we want to like find out what is our RNA-seq analysis and try to see it on the protein level. So
Hmm.
we stain the brain slices with few of the myelin markers and also few of the progenitor markers like the MBP,
Okay.
PTGF alpha,
Hmm.
CC1. These are the different myelin and progenitor markers. And
Hmm.
in each case, we are seeing that there is a difference in male and female.
Hmm.
So when we want to look into it, like what is the main reason, then we try to find out what are the other thing can contribute this.
Yeah,
Then
this.
we stain them for some axonal marker. There is the neurofilament. There is the kind of a neuron marker. And also we found the same thing, like the male mice are more like affected by the LB. So then we go for a more detailed imaging. Like we go for electron microscopy on the neuron, and we also find the same thing, like the control compared to LB, we
Hmm.
have a similar kind of trend. And in the male mice, it is more pronounced. So actually, to get an idea why this is happening, we have to wait for a few days because the sexual maturity it comes or in the adolescent time point, which is after around P24, P24 to like around P30.
Okay.
So for that, what we take a strategy, like we want to inject a retrograde virus in the hippocampus, and we want to see what is happening like in the total brain areas. So a retrograde virus is a virus kind of like is a kind of tagged with a fluorescent dye.
Hmm.
When you inject into the brain, it travel back to the point where it comes from.
Okay.
There are a lot of interconnection between the brain regions.
Hmm.
So we inject in the hippocampus, in the SLM, and we see surprisingly, like there is a big chunk of like a red fluorescence. The actually
Hmm.
is tagged with red fluorescence dye in the cortex.
Okay.
In
Okay.
cortical part, so in cortical part, there are few different region. One is called LEC, lateral entorhinal cortex,
Hmm.
MEC, like medial entorhinal cortex. So we found that when we inject this retrograde dye, so it travel back to the medial entorhinal cortex and lateral entorhinal cortex. And we found a like in the LB and in the control, there is a huge difference. Like in
Hmm.
the LB, we found a very few number of cells that is stained with that like the retrograde dye. But in the control, we found a very high number of retrograde dye. That means the connection from hippocampus to entorhinal cortex is not well established in LB.
Okay.
And to check that in phenotypically, we use a behavior system that is called contextual fear conditioning.
Mm-hmm.
So, in this behavior, we put the mice in the first day. Like, if I say it is the time point where adolescent mice, you can say that is a adolescent mind. It's like P33.
Okay.
So now actually the sex come into the picture more pronounced. So when we did that behavior, so in the first place, yeah, if you want
Okay.
to,
Yeah. Yeah. Sure. No, I was gonna ask. Yeah, you started already, but I was gonna ask. Yeah, what was your main goal for this particular experiment? Contextual fair conditioning.
okay, yeah. So
Yeah. Go
that
ahead.
is
And you have a really interesting findings in that experiment. Yeah. Go ahead.
so in this behavior paradigm. Actually, if I like explain in a small time point, so it is
Mm-hmm.
very interesting behavior. So we put the mice in a cage.
Okay.
So in the cage, there is a context. Like inside there is a box. Like there are some bars, there are some holes. So we put the mice inside that, and there is some shock event after like 5 minutes, after 6 minutes, after 7 minutes. So
Okay.
we keep it for like 13 minutes on that specific box.
Condition. Hmm.
Yeah. And then we remove the mice from that box. And on the next day, we put the mice in the same box, but that day we are not giving any shock.
Okay.
But interestingly, on the 5 minutes, like the first shock point, mice freeze.
Hmm.
So they have a memory that on that time point, they're gonna experience some shocking, but there is no shock. In
Okay.
case of control, they have a better sense of this context. Like
Hmm.
we will get there, and after 5 minutes, we'll get a shock. But in case of LB, especially the male LB,
Okay.
they don't care. They are moving around. So even in the second day, when we put the mice in the box, they're moving around. So they don't think of that like they might have a shock. But in case
Okay.
of female, they know they are like behaving as the same as the control.
Yeah, so that means, in a simple way, male mouse,
Mhm.
they lost their sense
They
of fear.
lost their sense. Yeah.
Okay.
This is called a contextual memory.
Hmm.
So they cannot remember the context.
Right, right. So,
So when we want to look back that what makes this context memory, then we found the cells in the LEC or lateral entorhinal cortex or
hmm.
MEC. There are a few cells that is a reelin. These cells are important for this contextual memory.
Okay.
And when we stain them for the reelin, and we found that in the LB, the reelin positive cells are very low,
Okay.
especially in the male.
Hmm.
So that means like they have a developmental error from the very beginning of their life. So their LEC is not well developed. That's why the projection from LEC to hippocampus is not well established in LB. And
You mean dorsal hippocampus, right?
dorsal hippocampus, yeah.
Mhm.
So uh, this projection is very important in long-term memory, also the contextual memory. So that's why in the behavior, the LB male mice cannot remember because their like the projecting cells is not there and the projection is not good. So to cross refer this thing, we did like diffusion MRI. It's a kind of MRI based on water movement in the brain. So
Okay. So is it
this,
the same MRI that we do for
yeah,
human?
in human. So it
Okay.
is a very interesting idea. Actually, it's a very
Hmm.
translating the research. Like we want to translate the same thing what human did, and in like in mice, we are doing the same experiment and find the similar kind of results actually. So there is a study that in TBI human, like those who experience a traumatic brain injury in the childhood,
Okay.
and in the regular child, they had a MRI study, and there they found this entorhinal cortex is like there is a less connectivity in the entorhinal cortex to the hippocampus, dorsal hippocampus, in the traumatic brain injury children than the control group.
Okay. So
We found a similar result in LB, that there is a connectivity problem
mm,
in the dorsal hippocampus and the entorhinal cortex in the mice.
okay, that's so interesting. So if you extrapolate your observation in mice to
Mm-hmm.
human,
Mm-hmm.
uh, another group also previously studied this, observed the same thing that's happening there.
Yep. Yep.
That's so interesting. Tell us about your study. Like, what was kind of the most challenging part of the study, and how did you solve that problem?
So actually, when we are trying to do the stereotactic injection into the dorsal hippocampus, so actually there is no study. I search a lot. My supervisor also search a lot. We cannot find any specific surgery protocol that inject on the specific area where we are focusing. And for stereotactic surgery, it is very challenging. Like if you move a 1 micrometer, so it change a lot.
Okay, it has to be exactly.
It has to be very precise.
Okay, okay.
So I'm trying to find out the exact coordinate for like 2 3 months. One
Hmm.
day I was having my lunch at the lunchroom and I had an orange on my hand. So I thought like, why don't I draw the thing here? Because it is a 3D model. So our brain, like the mice brain, is the same as the 3D. So I draw the lambda, bregma, and then z-axis. Then after that, I found actually like the exact point where we should target.
Okay, so that's so interesting. Here's the experiment. Yeah, go ahead.
Took the picture. Actually, it was a one kind of important aspect of my study. So if you see on the left side, so we are focusing this red area. So we want to see this red signal here. So previously, when we inject in the dorsal hippocampus, we are not getting any signal in this area. So it was a nightmare for me for like last 2 months. I was working, but after this, I got it. So it was one of the biggest challenging part of my study. Yep.
Okay, that's interesting. So you kind of did that for the first time, this surgery.
Yeah, yeah.
Okay.
And right now, our lab is working on different projects, and they are using these coordinates for their reference.
Hmm. Okay, I have a kind of philosophical question.
Mhm.
Um, so, in evolutionary perspective,
Okay.
what's the what, what's the importance of these sexual dimorphic changes? You think, in
Okay.
your opinion? So I'll give you one
Yeah. Yeah. Yeah.
example. So it's not only this brain development. I
Yeah.
was just reading one article published in an special issue of Science Journal this
Mm-hmm.
August. So there they said they showed the sexual differences in men and women
Mm-hmm.
for immune system and their immune responses.
Okay.
Like men are more vulnerable for viral infection, acute infections, and
Mm-hmm.
some cancer uh,
Mm-hmm.
due to some cancers. But women's are more protective. I'll say their immune system is much better in
Better.
those situations.
Yeah. Yeah.
But there is a but. The situation women may have
Uh-huh.
over immune response, so
Okay.
that's why sometimes they have autoimmune diseases
Autoimmune disease. Okay.
over responses.
Mm-hmm.
So in immune system also men and women are different,
Yeah.
but so that's why I'm curious. So what's the evolutionary advantage or disadvantage of these differences between sexes?
Yeah. So, uh, in common criterias, male are more vulnerable for any kind of stress, any kind of disease situation
Okay,
because of
you're talking about human or mice, both?
human,
Okay.
human,
Mhm.
because their growth and exploration make them more vulnerable.
Okay.
In case of evolutionary aspect, men do the most of the exploring things. They encounter more challenge, more stress. So that's why environmental instability is there. Like they have instability inside their blueprint.
Okay. Okay.
So that makes specifically in this mm, like uh, sexual dimorphism in the stress response.
Mm.
So we can say that there is a reduced neurogenesis, a reduced myelination, which is a kind of a trade-off between early independence and long-term cognitive flexibility. So it is little complex actually. So in long term, sometimes this stress can help you to shape your next endeavor. Sometimes this early stress can lead to you a destructive situation of your life. So
Right.
you cannot say like this is the
Yeah.
only reason. If we had a stress in the early stage, this will definitely happen. No, you cannot say. It might happen, but there might be something other happening. In case of
Mhm.
female, they have a resilience. Or what are the resilience? Like they have a more robust neuroprotective mechanisms. They have a enhanced neurogenesis. Mainly the thing what I think mostly working, that is the hormone buffering.
Okay.
and they have a caregiver philosophy in their blueprint. So
Mm-hmm.
that makes their social memory more strong.
Okay.
That's why they can take more stress, they can bear more problem. But as you said, that might sometime backfire.
Yeah. I like the term you used as the trade-off. So
Yeah, so it's a kind of trade-off. So
yeah, yeah.
you
Sometimes
can,
it is good, yeah,
so
for one
trade-off
thing.
is a common term in evolution actually. So
Mm,
when you get one, you will lose one.
right.
Yeah.
Okay. Thanks for the explanation. And so my last question. So what is kind of the future direction from here for your particular projects? So what can be done next?
So our next plan was to start these projections from different time points. Like we want to see in which time point this disruption is happening.
Mm-hmm.
So like we checked it on P33, that is like 33rd days of the mice. So
Mm-hmm.
we want to assess this in different time point like P5, P10, P15,
Okay.
and we
Okay.
want to see it in like you can actually genotype them for the sex in the early age, like around P7. You cannot see
Mm-hmm.
anything, but if we genotype, we can say that this is XY or this is XX.
Okay.
So we can see that, like, is there any difference in this male and female in the early stage, and what are the difference? So for
Mm-hmm.
that, like, you have to inject the viruses, dread virus. So we can say this is a chemogenetic activation. So we'll inject some viruses, and after few days, we will inject CNO. CNO is a kind of modulating molecule that will bind to the gene uh, and activate some fluorescence inside the mice model.
Okay.
So this is a little bit deep. So I will not go through like that deep.
Mm-hmm. Yeah.
Yeah.
Yeah. Yeah.
So
Sure.
that is our main target. Like we want to find out the exact time point, where this specific lose of the connection is happening, and why this renin positive projection from the entorhinal cortex to dorsal hippocampus is happening in LB, specifically in male mice.
Okay. Okay. I hope this study will be successful and we'll know much more about that.
Yeah.
Okay. So finally, do you have any take-home message for our audience?
Yeah. So as I said, um, sometimes like as we work on stress for like few years, as I see, not always like the stress is bad.
Hmm.
So sometimes this stress can shape your brain more perfectly. As I said, like in the early stage, the synaptic pruning is a very important aspect, and stress speed up this synaptic pruning in some point. So
Okay.
there is a threshold. So stress, like maybe if you put it too much stress, that might hamper your connectivity in the brain.
Hmm.
But if some kind of small stress, that will boost up your plasticity of the brain.
Okay. So
So
if I
for
just
kids,
paraphrase what you're saying, instead of stress, if I say if we take challenges,
mhm,
um,
yeah,
it will help us to grow better,
to grow
like
better.
up
Yeah,
to a stage. Okay.
yeah,
So
yeah,
kind of because challenge give us stress
yeah,
sometimes.
yeah, yeah.
Okay, that's a wonderful message. Thank you, Rafiat Bhai,
Yep.
for explaining your research so wonderfully, and also for coming to this platform. Thank
You're
you,
most
audience. Thank you,
welcome. We had a great time.
and see you again in the next episode. Bye.